I'll turn it over to you first, once we go through the introduction and executive summary. Let's give it another minute or two to see who else pops in.
Hi everybody, this is Manuela Basso, and I have Francesco's computer because he will present the data then later on PRMT6 in vivo, inhibition in vivo. Hello.
Okay, great. So we have you up on the agenda after Sam and Michael. So I think we can probably get started. Yeah, so in terms of agenda, I'll just go through a bit of a summary of what's gone on since our last meeting about a month and a half ago. And then I'm going to turn it over to the law students, Michael and Sam, who are going to go through their analysis on the freedom to operate around our main lead series. And then we'll turn it over to Manuela's group, and she's going to give a presentation on PRMT6 inhibition in an in vivo model for SBMA. And then we'll finish up by going through some of the new compounds that have been prepared by the UHN team and a little bit of methyltransferase data. So in terms of what's gone on in the past month or so, the UHN team submitted 19 new compounds for primary assay testing, and they have about another 9 that are close to being ready to submit it. They're just going through QC testing. For modeling, Conrad conducted an exercise where he kind of looked at that lead series on the C ring, where we've been spending most of our efforts recently, and looking at what would happen if we removed the carbonyl group and replaced it with a directly linked ring system, and see if we could replace that carbonyl hydrogen bond. And from his modeling exercise, it looked like that was possible. We could get a different ring system, which would hydrogen bond to the same backbone that the carbonyl does. However, we saw a lot of steric interaction between that modification and the B ring. So created a lot of torsion, basically puts the molecule, then doesn't bind properly, so we get quite poor docking scores. So based on those modeling results, there's really no rationale to pursue that type of modification. Feng Ling has tested 8 of our, I guess, most potent compounds in an extended panel of methyltransferases, and they were all very selective. So not a huge surprise, but obviously we need to generate that data. And just to finish off the summary, Rachel went to the KDA conference last week in Orlando and presented a talk on the program. She's not here, unfortunately, today. She's got other commitments, but she did tell me that the talk was very well received, and she had a lot of comments from both academic investigators and industry. But I think what thing that was nice to hear was the patient groups were particularly, I guess, excited and impressed with how we've built a collaboration and the fact that we're the willingness to share our data openly with the community. So overall, I think it was a very, very positive that we had a chance to present our work in front of some world experts in the field. So with that, I'm going to stop sharing my screen and turn it over to Michael and Sam.
Great, thank you, Peter. Just share my screen. Okay, does this look good to everybody?
Yes.
Okay, perfect. Good morning, everyone. My name is Michael, and today with my colleague Sam, I'll be presenting the results of our PRMT6 Freedom to Operate analysis. So just a bit of a roadmap for the presentation. I'll describe what the Freedom to Operate analysis was and why we performed it. I'll describe our research methodology for it, and then Sam and I will go over our results. So the purpose of our Freedom to Operate analysis was really to determine the outer bounds of how we can modify a chemical starting point, in this case SGC 6870, using medicinal chemistry to create a drug-like molecule. So I'm sure most of you are familiar with this structure. It's here on the right-hand side. And the question that we're asking really is, are there any intellectual property claims that might limit our ability to use medicinal chemistry to create a drug candidate from SGC 6870? So the way that we go about doing that is by identifying existing intellectual property claims, for example, by active or pending patents, for compounds that are structurally similar to SGC 6870 and that are being used for similar purposes. And so in doing this, we keep in mind the three areas of planned exploration on SGC 6870, the A, B, and C rings. So because A ring binds a solvent accessible pocket, we're looking at modifying that with solubilizing groups. So really what we're looking for is any patents which claim benzodiazepinone cores with those kinds of modifications on the A ring that might get in the way of our exploration. Similarly, we're looking at modifying the B and C rings for potency and ADME. So we want to look for any patents which, again, claim a similar core with different modifications on the B and C ring that might get in the way of us making those modifications and optimizing for potency and ADME. So the methodology that we used was twofold. We performed two different searches, a structural or a Marcouche search, and then a text search. The structural search is a typical query, but instead of using text, we're using chemical structures. We use a website called SciFinder, which I'm sure many of you are familiar with. And what you do is you search for SGC 6870-like chemical structures, and then identify any patents which claim those compounds or methods which employ those compounds. The text search, we use lens.org, and that's just a typical text query. On that site, we searched for the structure itself, SGC 6870, and then all the different aliases of PRMT6, since we all know that genes and proteins can have a million different names. And from that search, we identified patents which either claim compounds or methods related to SGC 6870 or PRMT6. I'll turn it over to Sam to discuss the Marcouche results.
Thanks, Michael. So we began our search with a Marcouche search. It's a broad structural search based on the substructure of the substance, and we are searching to see whether our compound is covered by broad structural claims. And we're trying to see if there's any patent that covers the SGC 6870 and or would cover any potential modification we want to make that could potentially come into contact with any existing patents. So we began our search with the exact SGC 6870 structure, and then we slowly broadened the search scope by allowing for increasing structural variations. And we did this until we basically got just a benzodiazepinone base, which returned essentially all patents claiming some kind of benzodiazepinones. We restricted the scope of our search to just Canada, the US, the EU, Europe, and WIPO. And we also limited our search to patents filed since 2005. We did this because patents have a 20-year limit, and we really only care about unexpired patents. All of this, we got 83 results. So then we took the resulting 83 patents and did a preliminary review for relevance. We found almost all of them referenced some kind of existing commercially available compound, such as Xanax or Valium, which were captured by the structural search. We found that actually very few of the claims were structurally related to the molecule itself, and we ranked all of the results on a 1 to 3 scale based on their relevance to us. In total, we found 75 results to be irrelevant. We found 8 results to be somewhat relevant, and then there were 0 results that we found immediately that we believed to affect SGC's purpose. After that, we followed up on some of the results that showed interest, and then upon further review, we found that all 8 results were found to be actually irrelevant for the SGC's purpose, even though it included some kind of a structurally similar claim. So here we have one of our more interesting search results from the Marcouche search. Here we have a patent filing for bicyclic 1,4-diazepinones and therapeutic uses thereof. It's filed by Cytokinetics, which is a US biopharmaceutical company. They're actively pursuing this patent family, and there are 2 patents granted in the US and patents pending in Australia and Canada. Upon first glance, the patent gained our attention because it seemed to cover the bicyclic 1,4-diazepinone backbone of the SGC molecule. So then we, upon further review, looked at the specific claim language of this patent. So we found that this patent wouldn't actually cover SGC 6870 for 3 reasons. So first, SGC 6870 has a methyl group at RY, which you can see at the leftmost end of the molecule. And at that position, the patent doesn't claim a methyl group, whereas SGC 6870 has a methyl group there. So the SGC 6870 wouldn't fall into that patent. Second, SGC 6870 doesn't have a functional group at R1, which is on the right most side of the molecule, and the patent requires a hydrocarbon chain at that position. Lastly, SGC 6870 has an arrow group at the R3/R4 position, and the claim doesn't allow for any arrow groups at that position. So on the next slide, we have drawn the closest molecule to SGC 6870 that could potentially be captured by this patent. And then we kind of did the overlay of the requirements. And you can see exactly what I just mentioned. At each position, there's a requirement that the SGC molecule doesn't meet. And turning to the last slide, for the Marcouche search, we can compare the SGC 6870 molecule to the closest molecule that will be captured under this patent. We see that the SGC does not intend to modify the R1 position to include a hydrocarbon chain. So any planned variation of the SGC molecule will not be captured by this patent. And also, even though R3/R4 are in the B area for planned exploration and modification, we don't really anticipate that it will change from an arrow group to a non-arrow group. So the SGC is essentially free to explore any modifications on the B ring without having to worry about infringing upon this patent. And I will now pass it back to Michael for text searches.
Thank you, Sam. So with respect to the text searches, we got 3 general categories of results. First, there were a number of patents which claimed methods of inhibiting arginine methyltransferases like PRMT6, but for the treatment of diseases aside from SBMA. So there's a number for cancer, clonal hematopoiesis, and ALS. But again, because these are targeting other diseases aside from SBMA, they're not really of concern for us. The second general category that we had was patents claiming diagnostic methods where detection of PRMT6 expression is used as a biomarker, again, directed at purposes not related to SBMA, so not really of concern for us. And then finally, we found one patent which claimed a method of treating SBMA using a combination therapy of a PRMT6 inhibitor and an LSD1 inhibitor. So this, of course, was of interest to us because it specifically mentioned SBMA. So I'll talk about this patent in a bit more detail and refer to it as the 525 patent. So the title of this patent is LSD1 inhibitor and PRMT6 inhibitor for use in the treatment of a disease associated with gain of function of androgen receptor and or with overexpression of an androgen receptor coactivator. So of course, as I'm sure everybody here is aware, both PRMT6 and LSD1 are coactivators of the androgen receptor, but we're particularly interested in PRMT6. So the applicants on this patent are nonprofit organizations and research institutions out of Italy and France. But what does this patent actually try to claim? So it's claiming combination therapy for treatment of SBMA, consisting of both an LSD1 inhibitor and a PRMT6 inhibitor. So most of the claims refer to genetic downregulation, so using RNAi or antisense oligonucleotides. And important to note is that the patent claims specific sequences for each of these purposes. A few of the claims of the patent do claim small molecule mediated inhibition of LSD1 and PRMT6, but really importantly is that they don't actually claim any specific compounds for this. They just make a general claim to LSD1 inhibitors and PRMT6 inhibitors, small molecule inhibitors, I should say. So the current status of this patent is that there is a European patent application pending, and there is not currently any published application in the US or Canada, but this final deadline has not yet passed for the organizations to apply for the patents in the US and Canada. So where does this leave us, and should we be concerned? Well, as a general point, these general claims to small molecule inhibitors, which don't actually claim any specific compounds, are often narrowed throughout the patent examination process. So our recommendation is monitoring the progression of the examination process in the relevant jurisdictions to see if this patent is narrowed down. Again, it's really important to know that this patent is only claiming combination therapy using LSD1 inhibitors and PRMT6 inhibitors, not PRMT6 inhibitor monotherapy. And so in conclusion, there's no big showstopper patent for our work. Generally, for the most part, there's nothing preventing exploratory modifications on the A, B, and C rings. Our recommendation is to monitor the progression of the 525 patent to see if it's allowed, if its claims are narrowed, and if applications are filed in the US and Canada. So with that, that concludes our presentation, and Sam and I are happy to answer any questions anybody may have.
That's fantastic. Really great. It's particularly that we have some freedom to operate around the lead series. I did have one question back to the cytokinetics patent. I don't know if you can maybe put your slide back up. It's particularly around the X1. Okay, so X1. X1 and X2 are independently nitrogen or carbon with an RX group. So this is a site where we could potentially modify and add a solubilizing group because modeling shows that that is actually a better vector to come off to reach out into a kind of a solvent exposed area as opposed to R1. And I'm wondering, in your opinion, if we were to remove that RY group, that methyl group, but then add pendants off of that X1 position, does that put us into their IP space? I mean, alternatively, we leave the methyl group in R1 if we have to, and then just modify off that X1 position.
This is Max. I would just suggest you go to the slide that shows the the SGC compound in comparison to the, maybe there is the most helpful spot. So sorry, go ahead. Maybe that's the best place to look at it.
Okay. So there's actually the patent for the patent to work, it has to fulfill all of the conditions. So even just one or two in this case that the SGC molecule wouldn't fulfill would basically put SGC outside the bounds of this patent claim. So even if you made modification to X1 or X2 in the arrow ring in the benzodiazepine core, it still wouldn't, and even if the first criteria is no longer met by the lack of a methyl group at the RY position, it's the SGC molecule would still not fall under the R1 position requirement, which requires there to be a hydrocarbon chain at the R1 position. And also there would still be an arrow group, the B modification area at R3 and R4, which would both put the SGC molecule outside the bounds of this patent, no matter what modifications you make to the X1 and X2 position.
Perfect. Thank you. Does anyone else have any questions for Michael or Sam? Francisco, you have a question.
Yeah, it's actually me. So it's not really a question, but it's more a point. So we are the group, Maria Pennuto and I are the group that apply for the patent, the last one for the combinatorial therapy. And you are right, we didn't claim any specific, actually that was a claim that at one point the offices decided to keep, but without talking about any specific inhibitor. Because when we try to patent the therapy only for PRMT6 and SBMA, they said that there was no novelty because we previously published a paper in which we show that the androgen receptor is directly modified by PRMT6. So we only went with the combinatorial therapy for this reason. And mostly we protected the artificial microRNA that we used in our therapeutic approach.
So if you get that combination therapy broad claim, including small molecule inhibitors, issued, and we want to do that in the future, we'll come and talk to you.
Yeah, exactly. And then we'll see also if we ever get to the other countries, because we opened to 6 different countries, but we still didn't receive any feedback. So it's all on the process now.
Are you planning to file in Canada and the US, if you can?
Yes, and in Japan and yeah, Australia, and then I don't remember exactly, because these are really things that are dealt by the Teleton office, so the charity that funded our project initially. So, but we can talk more about the details if you need. Yeah. The idea was, I'm not even sure if they already send the file the patent or they will, but the idea is to do that. But and I don't know in which form though. So
thank you.
This is all I can say because really I don't have other information. Yeah.
Okay. Well, amazing work, guys. Thank you so much. With that, I think we can turn it over to you, Francesco. You're going to present.
Yeah.
Hi, everybody. I'll try to share my screen. Okay. Okay, do you see my screen?
Yes, it's not in
Okay.
presentation mode yet, but
Yeah.
we see your screen.
Okay, perfect.
There you go.
Okay, I'll present some data about our preclinical trial. We try to silence PRMT6 in an in vivo SBMA mouse model. First of all, we use this AVMT, which is a muscle-specific capsid, which can express either a negative amir or a PRMT6 amir. And we do inject the spiral vector at 3 weeks old systemically. And then we start doing behavior test, or we have a cohort for dissection and histology. So first of all, we saw that upon this treatment, we were able to see target engagement in the quadriceps of these mice. So the PRMT6 levels are decreased in the treated group compared to the negative one. As you can see, wait, I use my pointer. So here, looking at RNA levels, we are still troubleshooting the immunoblot, so the proteins will come, but we see a target engagement at least at RNA level. Then we did the behavioral side of this project, and what we saw was an improvement in survival. So our mouse model is quite aggressive in pathology. So usually our mice die at 71 days old, so quite young actually. But upon PRMT6 silencing, we see a little bit of rescue. Actually, they die at 82 days old, so there is a 11-day improvement, which amount about 15% of the lifespan or more. So we are very happy about this survival improvement, but there are also other improvements. So we see an improvement in body weight. So do you see that there is a statistically significant difference in body weight from 8 weeks on? There is the negative mice, they lose quite a bit of weight and they do not look very good. And in contrast, PRMT6 mice, they do not lose the same amount of weight. And so this is quite positive, but also we look at the muscle side. So the quadriceps weight was also increased at 8 weeks of age. So there is also an improvement in muscle size and not just overall body weight. There are also some functional improvements. So we look at grip strength and hanging wire. So we measure the ability of mice to hang on this wire or to exert a grip. And we saw that although both the treated and the negative mice lost force, the PRMT6 cohort did much better in the grip strength test and also in the hanging wire, where you see that at 8 weeks of age, the negative group it's not able to hang for 60 seconds on this wire, while the PRMT6 group is still able to hang on. So we see a functional rescue in these behaviors. We also look at Rotarod. So this is a test where mice need to run on this cylinder, which accelerate with time. But in this particular test, we did not see any significant difference between the two groups. So this could also be due to the fact that we are muscle specific. So maybe we are not able to improve these skills because there is some motor neuron involvement in the pathology that we are not addressing with this specific treatment, which is mostly muscular. So to conclude this side of my presentation, the muscle-specific AAVs that express MR-PRMT6 treatment lower PRMT6 mRNA levels in the skeletal muscle. So we saw target engagement. And also there was an improvement in survival, body weight, muscle weight, and muscle strength. And right now we are doing histological evaluation. So we were doing hematoxylin eosin, NADH staining, and CSA analysis. So we look at the quality of the muscle, the fiber type, and the fiber size in the different mice. Then we also did some tests with PRMT6 inhibitors. We did some luciferase assays. So this is basically the system. We do transfect cells with a luciferase plasmid. So basically the system works as described in the image. So on the left, you can see that upon DHT induction, AR with PRMT6 translocate into the nucleus and binds to these RA sequences. And so the luciferase is transcribed, and then with the test, you will see some light. But when you do use an inhibitor, in theory, the PRMT6 won't bind to the androgen receptor, and so there will be no luciferase transcription. So we monitor how much signal we get from this luciferase assay. And first of all, we tested this inhibitor, EPZ020411, and we tested this in X cells, and we overexpressed PRMT6, and we also overexpressed the androgen receptors. And we did do 2 different tests, one with the wild type receptor and one with the mutated polyQ1. And we tested different concentration of this inhibitor, and we saw a dose-dependent relationship, both in the case of the wild type receptor and in the case of the polyQ1, as you can see from this graph. And maybe more of interest for you is also that we tested the SGC6870 and the SGC6870 NSA in the same conditions, so in X cells and overexpressing PRMT6 and overexpressing the androgen receptor that could be wild type on the left or polyQ on the right. And also in this case, we saw a dose-dependent relationship and curve, but what we did not see was that the negative was not truly a negative, so there was still some inhibition going on at the highest concentration, but we saw this inhibition both in the case of the wild type receptor and in the case of the polyQ receptor. So to conclude this part, at this range of dosages that were from 0.3 micromolar up to 30 micromolar, we saw PRMT6 inhibition in the overexpression conditions, and we were able to see a dose-response curve. But SGCN still seemed to have an inhibitory effect on PRMT6. So I don't know if, do you have any questions?
Well, I talked to Rachel about this data, and we were super excited to see that this chemical probe shows a dose response in your assay. So I think this is a really, really exciting result for the team, and I think it just gives us hope that if we, as we progress some more molecules and get better potency and more drug-like properties, that this is something that we can move forward with, and looking forward to certainly testing more compounds once we have some available in your assay. Yeah, I don't know how to wrap my head around the data with the negative control. I guess, is there a way to discern whether or not it's truly a result of PRMT6 inhibition, or is maybe that negative control doing something else that might pop up in your assay?
Maybe we can check also the viability of the cells
Yes.
at the same time that we didn't do. So I don't know. One possibility is that with the negative control, there is more toxicity, and so we lose more cells, and so the luciferase signaling is lower. This is something that we didn't measure, so we can add that.
Also, and
was that one dose dependent as well?
We have to test it.
Okay.
We will test the different dosages. Also, something that Rachel told me, but I'm not sure if I got it right, is that maybe the negative is like an enantiomer of the positive or some kind of version.
Yes.
So she told me that it could also be that shipping the compound, and maybe the compound is not so fresh. can they switch it and gain some like change form and go back to the like active compound?
Not impossible, but I'd be surprised if that's the case. I mean, certainly something that we could look at. It's been sitting in, was prepared a few years ago now. I would doubt that we're seeing racemization at that position.
Okay.
Were any plasma samples taken at all from the study to look at exposure of compound?
Sorry, I only understood the exposure, the last part.
I was just wondering if any plasma samples were taken from the mice just to measure compound levels.
So in the mice, actually, we didn't test these compounds.
Oh,
in
okay.
the mice, we did a gene therapy. So that was a proof of concept to understand whether removing PRNT6 would be really beneficial. And I have to tell you that in this mouse model that is super aggressive, we never saw with any other intervention this increase in survival. So it means that PRNT6 is surely relevant. And this is why I wanted to show you the data, because that could also be another reason for you to go ahead also with the compounds, because for sure, patients will never be compliant in receiving an adeno-associated virus injection while they are looking for inhibitors, pharmacological inhibitors. And I think that our data in vivo are simply a proof of concept that if you inhibit PRMT6, you may have a really beneficial effect in muscle. And we are thinking to then do the transcriptional the transcriptomic of this tissue, just to understand which are the pathways that really PRMT6 regulates and why we got this surprising survival. And I just want to highlight again that this AAV treatment was muscle specific. So here you are simply inhibiting PRMT6 in muscle is super beneficial. More than inhibiting everywhere, but maybe it's just a matter of target engagement with this compound, with this treatment.
So it was inhibited with the SGC compound. Sorry, I'm just trying to get my head around the study design.
Yeah, I don't think we never tried, because we received a very small amount. Like, I don't know, at least Maria Pennuto gave us a small amount to test only for the transcriptional activity. And so we never tried that in vivo, but we should consider together whether you think it could be interesting. I don't even know the pharmacokinetics or like, the toxicity, but or the dosage to use in mice. But for sure, it's something that we would consider of interest, either in this mouse model or in assembloids. So in models, in vitro models, where motor neurons and skeletal muscle cells are differentiated and put together in contact. And Maria Pennuto in Padova is developing these models as well.
Yeah,
understood. Thank you.
Yeah, we do have some PK on that compound. I mean, we do see exposure. It's not fantastic. I think there's a lot of work to be done there to improve PK properties around this lead series, but it's something we could potentially consider. But I think we have to see how much compound we have and whether or not we'd want to make more. I did have one question back with the antisense oligonucleotides you did. How do those oligonucleotides compare to the small molecule in your cell-based assay?
Yeah, that's a good question, because I'm not sure. Have we tried those?
We did
no,
trial
never.
Methyltransferase assay on
No, like we could try to do a comparison, because we never did a transcriptional assay with those. Maybe we did it like, really at the real beginning, but we were mostly looking at then the protein level, like, the decrease in protein level. And Yes.
Yeah, we look at protein levels, mRNA levels,
side by side.
not check
Yeah,
luciferases.
we could do that side by side with your essay, actually. Maybe in the C2, C12, perché questi sono per...
And which in your AAV experiments, which serotype of AAV do you use, and is it just a one-time injection, or is it yeah?
It's a one-time injection, and it is this capsid that's modified to be muscle specific. I think that the background is AV9, probably then a little bit twisted and modified to be muscle specific.
They call it MT, so we are collaborating with Geneton, that is a non-profit institute in France, and they use this, they called this virus AAV-MT.
Would the idea that if this goes into humans that you would do a repeat dose of this AAV? Or is it
one single, it's one single injection.
Okay.
Yeah, because of the possibility to hyperactivate the immune system and then have problems. So
yeah, I mean that's the big issue with AAVs is if you need to do repeat therapy, then you do get that immunogenicity response, and it kind of. Okay, now I think that was really fantastic. Really excited to see that data.
Thank you.
Does anyone have any additional questions? Okay, Frank, do you want me to present your slides?
If you want, either way is fine. It's a quick 2 slides.
Okay. You can see that, right?
Yeah.
Okay.
Sorry, did you want me to go?
Yeah, sure.
Okay.
Yeah.
Yeah, okay. So quick chemistry update from us. Like Pete said at the top of the meeting, we've submitted 19 compounds shown here. It's a combination of some of the proposed structures that were suggested via in silico screening, as well as some just ligand-based proposed structures as well, or analogs. And so, a lot of these we went back to these 5-member heterocycles in the C ring, as you can see, based on previous data that series behaved or performed kind of the best out of some of the modifications that we've done so far to date. Top row, CMD 2294 and 2295, you'll see that we've introduced piperazine on that A ring on the benzodiazepine. That's initially it was at the wrong kind of position. We are making the other one at the other position on that benzene ring for the solvent exposure kind of testing as well. And next slide. And we have about, I think, 10 compounds here, awaiting just QC, and they should be, we can submit these in our next batch of compounds. The top 2 compounds you can see, we've removed kind of the carbonyl group on that C ring portion off of the benzodiazepine, as well as the structure beside it. This is kind of our first analog where we're modifying the B ring structure. So it'll be interesting to see how those behave. We have planned as well to make the alkylated bromothiazine, just to have that comparison, that match pair comparison without the carbonyl group to SGC 6870. So that's in the works. And then you can see the rest of the compounds are just further modifications with the 5-membered ring heterocycles in the C ring portion as well. That's all from us. Any questions?
Yeah, I think it'll be interesting to see. That's the first analog we've had down in that B region. and according, at least predicting from modeling, this is really an area where it's worth exploring. Okay. If there's no updates or no questions for Frank, Fengling, I just took the slides that you sent me last week, so I think if you want to kind of just quickly go through them, I can just advance the slides if that's okay with you. Oh, I think you're muted. Oh, maybe we just can't hear you. No, sorry. No, I don't think so. Well, maybe I can just I can just go through. I think it's pretty quick. Like I said at the beginning, Fengling's group tested these 8 compounds, which are all either around one micromolar or less as the racemates, and testing them with related PRMTs 1 through 9. And as you can see, the percent inhibition of these compounds is really negligible. So we're not really seeing any activity with these orthologs of PRMT6, which again shouldn't be overly surprising since we are targeting the allosteric site. I think that is about all that I had for today. But I think I'll just open it up because I know that there's some people on the call that were at the KDA conference. If you have any comments about the conference or any interesting talks that were there, sure, Max, please.
Yes. Let me just put this. Hi, Francesco. We met Sunday's poster.
Yes, yes.
Very nice to see you again. Yes, so I was part of the KDA meeting, and since Friday, I pretty much went through the whole conference. It's my first one, and I was lucky, and then this also happened to be a very big one, and I was told by the chairman Terry from KDA, they have a big conference, international conference, every other year. And so not every year they have so many presentations and talks and posters, but this year happened to be a big one. So very, very informative, very educational. I met many people there. I listened to many interesting talks and many posters. And I think Francesco and Mariala's group, they have one presentation and they have multiple posters and in the PRMT and LSD1 target. So very interesting.
Yeah, just ask them if it's working. Ask if they can confirm if it's working.
The Peter already.
Yeah. So I think from my side, I don't have many comments. I'm still trying to digesting all the science, but very educational. I just want to provide some kind of update on the BD side. I think last time I might have mentioned I met a person in JP Morgan in January, and his name is Stephen Bailey. He worked with a variety of pharmaceutical company, including a Japanese pharmaceutical company, Shinogi, as well as a number of venture philanthropists, as well as a VC. And so I had follow up with him, but he just showed up in the conference, and very surprised. So I had quite a good conversation with him, and I had dinner with him, and we discussed many of the projects and presented in the conference. And then he actually very fluent in terms of the molecular biology and cell biology, and as well as the rare disease indication. We also connect to many people, and especially the projects in the let's say in the later phase, close to clinical stage. And he has a very high interest in that because those could be potentially bring into the attention of VCs and other investors. So I'm going to continue to engage with him, and he will continue to follow up with our project here. And I would say for Pete and Max, we probably need to bring him back in terms of the other Agora projects. And Stephen is pretty technical, and he understands a lot of the fundamental mechanism, and also he has a very big network in the VC side. So a very valuable asset. So that's all I want to say. Thank you.
That's great, Max. Thanks for following up with your BD contacts. Yeah, we should definitely have a discussion in the next couple of weeks on how we might be able to approach them and maybe get a meeting with them. Okay, does anyone else have any additional questions or comments? If not, I will start put out another meeting for the kind of the regular time, which I think is about the second last Wednesday of the month. And so I'll put that out for April, and we'll meet again. And by then we'll have hopefully some new data to share on the new tranche of compounds that were just submitted. All right. I really appreciate everyone taking the time this morning, and have a great weekend.
Thank you.
Thank you.
Thank you.
Bye, everyone.